We have been hearing so much about AI, transforming every field from healthcare to entertainment. But did you know AI's revolution has been proteomics too, especially in the structural biology? AI tools like AlphaFold3 are reshaping how we predict and understand the biomolecular structure. Curious how it works? It's styling.
Welcome back to Bioinformatics BB. This is Dr. Babajan Balanganapalli, Associate Professor in Bioinformatics. AlphaFold is an advanced AI model developed by a DeepMind that has revolutionized structural biology. It all started with the AlphaFold, which introduced the AI-driven prediction for protein folding. This was further improved with the AlphaFold2, which achieved a nearly experimental accuracy in predicting the protein structures. Now, AlphaFold3 taking things to next level. Unlike its predecessor, it doesn't stop at the proteins. It can predict the 3D structure of a DNA, RNA, and small molecule like ligands. These broader scopes allow it to model complex interactions such as protein DNA binding or how drugs are interacting with the proteins. In this video, I will walk you through what you can do with the AlphaFold3, how it works, and finally a hands-on demonstration. Let's get started. AlphaFold3 have many options to explore. The first one is predicting the protein structure. It will generate a highly accurate 3D model for proteins from single molecules to complexes. And second one is modeling the protein complexes. It will help us to study how proteins are interact with each other, predicting docking and binding interface. And third one is exploring the DNA and RNA interaction. It will understand how proteins binds to DNA or RNA to perform the function like gene regulation. And the fourth one is adding ligands and ion. It will help us to incorporate the small molecules and ions such as ATPs, calcium or magnesium or iron to explore their role in the molecular interaction. The last one is a post-translational modification. It stimulates the modification like phosphorylation or glycosylation and see how they affected the protein structure. So these features makes AlphaFold3 an unparalleled tool for exploring the biomolecular interactions. Let's see how the AlphaFold3 actually works and how it was trained. Let's break down how AlphaFold3 works using the protein as an example. Imagine protein has a long strand of bead on a string. Each bead represents an amino acid. The goal is to fold this string into a precise 3D shape that allow the protein to function. Here exactly how AlphaFold3 achieve this. Okay, so first step is single representation. Here the AlphaFold3 start by analyzing each bead individually, understanding the properties like the size, charge, and hydrophobicity. The second one is the pair representation. Here it will predict how the pairs of the beads interact even if they are far apart on the string. This step helps identify which part of the protein will come together when folded. So the third one is incorporating the multiple sequence alignment. The AlphaFold3 compares the input protein sequence to the thousands of similar sequences in a global database. This evaluation data help to identify the conservation and structural constraint guiding the folding process. The fourth and very important one is triangular attention. Using a method called the triangular attention, AlphaFold3 ensure all interaction follow the natural rules like maintaining the proper bond, angles, and avoid the overlaps or knots. The last one is denoising diffusion. Finally, AlphaFold3 refines the folding step by step, starting with the rough structure and interactively improving it. It adjusts the position each bead until the protein is folded into functional 3D shape. So finally, in short, AlphaFold3 combines the sequence data, evolution insight, and structural template to deliver the highly accurate protein structure for you. Okay, if you want to know more detailed analysis, you can refer to the article published in the Nature. And also, I came across the excellent deep analysis video on AlphaFold and AlphaFold3 by Dr. Mithuna Yoganathan on Looking Glass Universe YouTube channel. I will provide the link in the description box. Now, let's see AlphaFold3 in action. Here how we can use it. I have provided the link for the AlphaFold3 in the description box. You will need to register using your Gmail account and enter your academic details. Once that's done, you will redirect to the submission portal. Here, let's start with a simple example of protein modeling. First of all, you need to upload the amino acid sequence in the FASTA format. AlphaFold3 let you input sequence up to 5,000 amino acids, which is significant improvement over the previous tool that relied on Amenshow and homology modeling. But let me tell you, there is one limitation is that right now, you can only submit 20 jobs per day. To submit, simply paste the FASTA sequence in the input box, then click continue and preview your submission. You can even change the labels for your job if you want to. For example, here I'm using the CCR2 protein sequence. So within a few minutes, depends upon the size of protein, you will get your results. Now let's analyze the results. The first visual output you will see is the confidence score bar, that is predicted local distance difference test, PLDDT score. If you see the results is very high confident with greater than PLDDT score is 90, these regions are marked in a dark blue and indicate very high accuracy. If the confidence score is in between 90 to 70 region, the region is shown in light blue color and have a reasonable confidence. In the case of low confidence, you will see 70 to 50 PLDT score. This area are yellow in color and represents the low accuracy. Finally, very low confident, the PLDT score less than 50. This region often loops or a disordered part of the protein are shown in orange and highly uncertain region. The second output is the predicted align error PAE plot. Okay, this plot help assess the relative accuracy position between the residues. On the X and Y axis, you will see the residue of your protein, here the dark green region on the plot represent the low error, meaning high confidence in the residue placement. So the light green region indicate the higher error, showing the uncertain in the relative positions. Okay, these visual output makes easy to assess the reliability of your predicted protein structure and guide you for the further analysis. Now let's explore how to perform post-translation modification analysis using AlphaFold3. For this, you must need to know the PTM site in your input sequence. If you don't have experimental or literature information about the PTM position, there are so many tools are right now available for PTM prediction. It will help you to identify the sites in your sequence. Once you have this information, you can proceed with the following step. Okay, so first you have to go to the home page. First, we have to upload the amino acid sequence in the FASTA format. You can paste it into the input box, similar to the regular protein modeling. Once you have uploaded the sequence, you can begin adding the PTM. So post-translation modification are chemical changes that occurs after a protein is synthesized. This modification can significantly affect the protein function, stability, and interaction. In AlphaFold, we are having an option to select the two common types of PTM. One is phosphorylation and second one is the glycosylation. In phosphorylation, this involves the addition of the phosphate group to the amino acid like a serine and threonine or tyrosine. So we already know that one, the phosphorylation is majorly regulated the protein activity and play a key role in cellular signaling. And also we have the glycosylation option also available. This involves in the attaching sugar molecule, the glycans to residue like asparagine, threonine, or serine. Glycosylation also a very important step in the protein folding, stability, and cell communications in the receptors. Okay, let's add the PTM to the sequence. To add the PTM, click on the residue where you want the modification and select the desired PTM type. As I mentioned previously, you have an option to select only serine, threonine, tyrosine, asparagine residue for the PTM modification. Okay, once the PTM is saved, you don't have an option to edit the sequence furthermore. Make sure all modifications are finalized before saving. For example, in this case, I am selecting the serine, threonine, tyrosine, asparagine. Okay, so I'm selecting these 4 residues. If you scroll down to the bottom, you will find the option to select the PTM. For serine, we are adding the phosphoserine. Okay, for the threonine, we will include the glycan chain. In this example, I am selecting the NAG and MAN. This can read as starting the root with the N-acetyl glucosamide and add the branch of another N-acetyl glucosamide. And from this second branch, I'm adding the mannose. Okay, mannose branch. So you have to be very careful while adding this sugar molecule. You can refer to the FAQ section in AlphaFold for better understanding how the glycan chains, branch chains are added to your protein. So for tyrosine, again I'm selecting the phosphorylation. Finally, for asparagine, I'm selecting the hydroxylated PTM. After adding all the PTM, save the modification and submit the job. AlphaFold3 will generate the protein structure by incorporating all the PTMs. The time taken will also depend upon the complexity of the structure. Once the process is complete, you always have an option to download and visualize the structure of the PTMs also. Okay. At the end of PTM prediction, we can observe that the modification of residue shows slightly lower confidence compared to the normal protein prediction. Okay, this is due to structural complexity introduced by the modification like phosphorylation and glycosylation. So the PE plot also highlight the slightly higher error around the modified residues. This differs from the standard prediction, which usually have higher confidence and simpler structural assumption. PTM predictions provides a deeper insight into how this modification affects the protein structure and function. So this PTM analysis is the most powerful feature of AlphaFold3, allowing us to study the real-world biological modification and their impact on the protein structure. So these examples demonstrate the common PTM like phosphorylation, glycosylation, and hydroxylations. Remember, make sure to verify the FAQ for the fully supported PTMs and ensure the glycan structure are chemically valid to avoid that. Next step, we'll explore the protein-protein interaction and ligand interaction using AlphaFold3. So for this demonstration, I have chosen the hemoglobin protein as an example. Hemoglobin protein consists of 4 polypeptide chains, 2 alpha chains and 2 beta chains, along with the heme group that binds the action. So I am just including the sequence of 2 alpha chains and 2 beta chains. I am adding the 4 heme ligand group to stimulate the interaction in between the polypeptide chain and their heme cofactors. Okay, so let's submit the job. Wait for the model process and it will generate the results for you. So once the results are ready, you can analyze how accurate the heme groups are bind to each polypeptide chain. Since the structure of hemoglobin has already been well established, already it has been solved, already well documented in the protein database, you're likely to see very high quality of the PTM, okay, and IPTM. The result shows that very high confidence of most regions of the hemoglobin structure, indicating the dark blue is LD score is greater than 90 in the visualization. So the heme group bind accurately to the polypeptide chain with the IPTM score of more than 0.8 and a PTM score of 0.9, which reflecting the excellent quality. Okay, so PA plot also highlights a low error. You can see at very dark region here. indicating the precise residue positioning. So these high scores are expected as hemoglobin is well-studied structure or experimentally evidence available in the protein. For proteins or complexes without experimental evidence or homologous structure in the PDB, you might observe lower IPTM and PTM scores and more light regions in a PAE plot, indicating that higher uncertainty is there. AlphaFold3, however, still outperform any other computation. We could take the ITSR, Swiss model, Rosetta. It will provide an invaluable insight for the modeling and protein ligand and protein-protein interaction. So now let's explore the interaction between the protein, DNA, and ion using the AlphaFold3. For this example, I have taken the protein DNA ion complex directly from the AlphaFold3 example. Okay, so once I click on example, the setup has been included the protein sequences and the DNA forward and reverse strand sequences. Along with that, it added the two ions, the calcium and sodium. Okay, so once it is prepared, you just need to click on submit. So once the results are ready, AlphaFold3 provide detailed predictions of the interaction in between the protein, DNA. So similarly here, it is also going to show you the confidence score. It is more than 90 is there, which is darker blue in color, which is very high in confidence. Only few regions in cyan color is showing the minimal areas of moderate confidence. So the IPTM and PTM scores are more than 0.9 is there. You can see 0.93 and 0.94 is there, which is reflecting the excellent prediction quality. Even the PAE plot also predominantly dark green region, which is indicating that very strong alignment in the residue position. Okay, these results highlight the AlphaFold3 accuracy in the modeling, protein, DNA, ion interaction, especially on well-documented systems. So for next one, we'll analyze the protein RNA ion interaction. This is an example demonstrated the flexibility of AlphaFold3 modeling in RNA-protein complexes. So here also I'm going to take an example from the AlphaFold3 itself. So you can see here, I'm just clicking this example of protein RNA and ion on AlphaFold3. So the setups include the two protein sequences. So it's a dimer, two protein sequences, and here the one RNA sequence is there, and we are having the one ion that is a zinc. Okay, so once I click this one, this setup will show us how AlphaFold3 predict the RNA protein interaction and their associated confidences. So identically, here also you have this confidence score. Here also I got the greater than 90 scores are there. So here we got the structure include the mix of dark blue, cyan, and so most of the areas you can see that's greater than 90 is there. Some regions are the cyan also is there. So let's analyze the results for the protein and analyze the results for protein, RNA, and ion. So the confidence score, the structure include the mix of dark blue and cyan region, reflecting the very high content most of the areas. Some regions are in yellow color, corresponding to RNA flexibility or less defined interactions. Okay, and even the IPTM scores also slightly lower compared to the previous protein DNA RNA complex. Maybe this is due to RNA complex in nature. Okay, so P plots also, some regions are showing the light green region, suggesting slightly higher error in certain residue alignment. Okay, so while RNA protein interaction is inherently more challenging to model, okay, AlphaFold3 provides a valuable insight into this structural dynamics. Okay, this is all about the excellent prediction predicted by the AlphaFold3. Okay, before I conclude, let's look into the major pros and cons of this AlphaFold3. So thank you for watching. If you found this helpful, please subscribe to the channel. This is Dr. Babachan Vandanpalli and you are watching Bioinformatics with PEV. See you next time.